Hello everyone, welcome to SUNYA IAS, and this is your what to read and how to read video for Prelims revision program, also called as SUNYA Super 50 batch. And in this video, we are going to talk about that what to read and how to read for Science and Technology test number 2. Also, before beginning this video, I would really want you guys to please comment below and tell us that what are your reviews about this program and are there any improvisations which you guys want us to make. All right. So let's start. With respect to this test, you have to study the concept of genome, followed by the concept of genome sequencing. We already did in class that what are the all projects running with respect to genome sequencing at the international level. Then we did them at the national level and followed by the Department of Biotechnology and the Ministry of Science and Technology. So we did all those 6-7 initiatives, but the conceptual part was left. So just understand what is genome and what is the concept of genome sequencing. All right. Then move on to the concept of DNA profiling and compare it. What is the difference between DNA profiling, DNA fingerprinting, etc. Then move on to the concept of gene therapies, जितनी भी news में है, do the current aspect as well. Then there was this concept of dark genome in news, so read that. Then do the difference between genome sequencing and genome mapping. Then move on to the genome sequencing project. This is already covered, all right. So this you can revise from today's class only, and in tomorrow's test they'll be asking it, all right. Then few consortiums. There were in total 3 consortiums in news. Basically they were formed by DBT during the time of COVID and post COVID. So do that 3 consortiums. Then do the process of genetic engineering. What is the entire process? What is the function of nucleases? What is the function of ligase? When we'll be using it, and in this. Nucleus is also what are endonucleus, what are exonucleus, all these concepts have been done by you. Then you have to do what is the difference between genetic engineering and transgenic techniques. Move on to the gene drive technology. Then do the genetic engineering versus hybrid, in दोनों में difference करना. ठीक है, this already we have touched this aspect in the environmental classes, but again for the purpose of science and technology you have to do that. Then what is the regulation of GM food and GM crops in India? Then in the genetically modified crops, these were the crops in use, so you have to do them in detail. Follow that is BT brinjal, golden rice, wheat, Thara mustard, BT cotton, and BT rubber. And few miscellaneous were also there. Example, flavor saver, the first genetically modified tomato, right? So that was also in use, so do that as well. Then there was this food code in use called as Foodex Elementaries. You have to do that. Then you have to do the difference between DNA and RNA. What is the concept of gene expression? What is the phenomenon of RT-PCR? What is RNA interference technology? What is recombinant DNA technology? What is this concept of CRISPR-Cas9? And followed by the mitochondrial disorders, example 3-parent baby, pro-nuclear transfer. What are these? All right, chalo. Then you have to move on again to the concept of gene expression and transcription, which you have to do from the perspective of immunity. ठीक है जी. Then we'll start immunity, and immunity में you have to do what is innate immunity, followed by acquired and adaptive immunity. What are the innate that are the physical and chemical barriers present, for an example inflammation, interferons, tears, stomach acid, natural killer cells, and saliva. That all you have to do. Then you have to move on to the acquired or adaptive immunity, right? इसमें you have to do the B cells and T cells, antigens and antibodies. What is primary response and what is secondary response? इनमें do the comparison, which will be strong, which will be weak, which will be developed earlier by the body, which will be developed late by the body. And finally you have to refer to humoral or cell mediated immunity. Then you have to do the virus. इसमें you have to do the viral test, diagnostics test, what are the antibody test, and after these tests you have to do the vaccines which were in use. This has been a previous year question as well, so again we cannot skip this aspect. You have to do live attenuated vaccines. Also one important aspect is the example, so do examples of all these vaccines. ठीक है, तो सिनोवैक किस कैटेगरी में लाई करेगा, भारत बायोटेक किस में लाई करेगा, मॉडरेना will lie under mRNA vaccines, this and that you have to do. Inactivated viral vector, subunit vaccine, nucleic toxoid vaccine, etc. Then you have to do the concept of recombinant vector vaccines, followed by the diseases. इसमें chart बना लेना that what are the diseases which have been eradicated nationally and internationally. So we already have done in the current affairs class, so do it from there. Then if you talk specifically about diseases, you have to cover HIV, tuberculosis, influenza, polio, malaria. And while doing malaria, do the difference between this mosquito called as Anopheles and Aedes aegypti, which are the diseases which are caused by Aedes aegypti and which is a disease which is called by Anopheles. And also do the physical features of that their area of dominance, which is the part of the day attack and all. This is the aspect which is remaining, and UPSC can ask you. Then you have to do Kala Azar, lymphatic filariasis, hepatitis, neglected tropical diseases, antimicrobial resistance concept. Then moving on to the drugs. Drugs are also divided into Schedule H and Schedule H1 categories. So उसमें वो दोनों देख लेना. Then what is the concept of fixed dose combination? Then you have to refer to the one health principles, also called as Manhattan principles. ठीक है, तो ये देख लीजिएगा. Then what is the concept of zoonotic diseases? And lastly you have to do the alternate systems of medicine. In this you have to do Ayurveda, Yoga, Naturopathy, Unani, Siddha, Sovarikpa, and Homeopathy. For all you have to do like in crux you have to do all the ones for which this Ministry of Ayush has been created. Alright, so this is with respect to the second test. All the very best, guys. I hope you will revise enough, and give your best in tomorrow's test. Chalo, see you guys.